The cardiovascular system is at the heart of all things A&P, pun intended. So in this video, we are going to go through a comprehensive overview of everything you need to know about the heart. So stick around for the whole thing because I'm going to give you some practice test questions that I know your instructors are going to ask you if you're taking a test for the cardiovascular system. Hey everybody, Organized Biology here, where we make difficult biology concepts simple. And we're going to start with the cells of your body because that is the functional unit of life. You have 30 trillion of these very needy cells that I always think of these little baby birds that are like constantly asking their mothers for food. And so you've got these spread throughout your body and you need to feed them. Well, what do you need to feed them? Well, a variety of nutrients, oxygen, the gas from the air. You need to give these things to these cells in order for them to survive. Now, how do they get that, obviously? They get it from your bloodstream. So in order to get blood circulating throughout the entire body, we need the cardiovascular system. So if you look at the C, the word cardio always refers to heart. Okay, so cardio, heart, like if you're doing cardio exercise, right, you're working out your heart muscles. And then you also have the vascular part of that word. Vascular just refers to the vessels, okay? So basically the tubes with which the blood is running through. Now, I'd just like to think of this as the pump and the pipes. So you've got pump and pipes for specifically blood. Now, what did I mention that these cells need? Well, one thing these cells need is our friendly molecule of oxygen, okay? Why do our cells need oxygen, O2 gas? Because in order to produce the very, very important energy currency molecule ATP, we require oxygen as well as glucose, other nutrients. But we're going to focus primarily on oxygen. So we need to get this oxygen in the blood to these cells. How do we do it? Well, we look at the heart and we see that there's kind of a division in the heart. On the left side, be careful there, if you're looking at it, this is the left side of the heart. I'll write it over here. Let's go right there. That is the left side because it's as if you are looking at the person's heart. And then on the other side, we have obviously the right side of the heart. So that being said, look at the left side. We see it's very red. We know that red always indicates high amounts of oxygen. So I'm just going to write high O2 in red. And then obviously we're going to counter that with on the other side, very low O2 in blue. Now the left side of the heart deals with carrying oxygenated blood. So I'm going to run through this. I'm sure if you're watching this video, you've kind of been taught the cardiovascular system before. So I'm going to run through the structures of the circulation of the cardiovascular system itself first. So we've got the left atrium here, and that left atrium is going to contract. So I always think of the heart pumping in this manner. You take your hands and you contract the atria first, and then we're going to contract the ventricles. So this is the heartbeat. You can do it with your hands, and that's how the heart's going to beat, right? Atria, ventricle, atria, ventricle, atria, ventricle. So atria contract, push blood through this valve. This is called the either mitral valve, left AV valve, or the bicuspid valve. So there's three names for that valve, preventing backflow of blood back into the atria. It's going to get to the left ventricle, very strong, meaty part of the left side of the heart, and it's going to contract to the moon. Always think about it this way, right? We're contracting the left ventricle, and it's shooting, quote unquote, to the moon. That's because there is a semilunar valve that it's always going to go through. So whenever the ventricles contract, we're shooting through the semilunar valves, okay? Lunar means moon, so we're shooting up to the moon. In this case, number 4 would be the aortic semilunar valve because we're going to be traveling through the aorta, number 5 next. Now from there, you see the aorta is this massive artery carrying a lot of oxygenated blood up to the higher parts of the body above your chest and also your arms. And then we also have the descending aorta going down into the thoracic abdominal cavities and feeding the cells of the lower body and all the internal organs. So very important. We get that blood, the high oxygen blood, to the cells. Good job. So the oxygen will go from the blood in high concentration, flow high to low into the cells, and the cells will produce ATP from that. So we'll see that on both sides. Oxygen high in the blood going into the cells. Now the location at which this is happening is going to be called the capillaries. We're going to go through those a little later. Capillaries. Okay, very tiny blood vessels. They have fenestra, these openings to allow things in and out. So we feed the cells oxygen, we make ATP, but as a byproduct, interestingly enough, our cells will produce a gas called CO2. So I'm going to draw CO2 that's produced, and it's going to throw that into the bloodstream. Okay, that's what the cells do. They produce CO2. They're like little smokestacks, putting it into the bloodstream. So now we have low oxygen, high CO2 in the blood. We're going to determine that this blood is now slightly blue, although it is not. Okay, so we have slightly blue blood. Where do we need to get that blood in order to get oxygen again? So we lost oxygen, right? So think of this, low oxygen in the blood now. Where do we need to go with it to basically gain oxygen and then basically lose CO2 out of the blood? Well, you can think we need to go to these structures called the lungs. So you have a left lung over here, left lung, and then you have a right lung over here, right lung. Now, in order to get there, we need to pump the blood there, right? So think about it. Once we feed the cells of the body, we need to go back to the pump, right? So we can pump it to the lungs. The way the heart always beats, and I want you to remember this, you can say this over and over again, heart to body, heart to lungs. Heart to body, heart to lungs. Heart to body, heart to lungs. That is how we circulate. We went from heart to the body. Now we're going to go back to the heart, and then we're going to go to the lungs. Heart to body, heart to lungs. So we're coming back to the heart then. Where do we go through? Well, we come through either one of two vessels. If we're on the top side, we go through this superior vena cava, the top part vein cave, right? So we're coming back to the heart. Same thing here, we've got the inferior vena cava going back into the heart. Now, which part of the heart are we going to go into next? Well, we always start in the atria first. So you can think A is the first letter of the alphabet, and then we're going to go to the ventricle afterwards. Okay, so we go to the right atrium. Right atrium contracts again, goes through a valve called the tricuspid or the right AV valve. To prevent backflow, go to the right ventricle, the bottom part. We shoot blood to the what? Pulmonary semilunar valve. Remember, pulmonary, anytime you hear the word pulmonary, it's going to mean lungs. So through that pulmonary semilunar valve, we go to then the pulmonary artery. Anytime you hear artery, you've kind of noticed, it means we're going away from the heart. Whereas anytime I've mentioned the word vein, we're always coming back to the heart. Okay, easy way to remember it. Artery away, vein vacuums back to the heart. So we go through the pulmonary artery, we're going towards the lungs, and what will happen is, again, we have low oxygen in the blood, but we also have high CO2 in the blood at this point, yes? So think about it. In the lungs, we're breathing in air that will be very, very high in oxygen. In the air in the surrounding part of our body has very, very low amounts of CO2. So think about it. We have this concentration gradient where things want to flow high to low always. So where will oxygen go? Oxygen will flow high to low into the bloodstream. CO2 will flow high to low into the lungs, and we will push that out, breathe it out. So we now have very oxygenated blood that we have just received from the lungs. That's fantastic. So this happens on both sides. Now think about it. We just went to the lungs, we picked up oxygen. Where do we need to go back to? Heart to body, heart to lungs, and then back to the heart, right? So notice, these veins will bring blood back to the heart in this direction, okay? So this structure, number 13, would be the pulmonary veins. We have them on both sides, obviously, because we have two lungs. What am I saying? Two veins. We've got two lungs on both sides, so we're coming back to the pulmonary veins, and we're going to finish up at the left atrium where we started. And I realize I didn't draw this last one here, okay? So that is the circulation of blood, again, from the left side of the heart to the body. Feed the cells, come back to the right side, pump blood to the lungs, pick up oxygen, drop off CO2, come back to the left atrium, and we repeat over and over and over again, about 60 to 80 times every minute, because that's how often the heart beats. Now, that's important in terms of the circulation, but we also need to know the layers of the heart. What parts of the heart are actually doing some work for us? So I've zoomed in, so this is the heart again, to the layers of the heart. So we're taking basically a chunk of the heart, and we're going to zoom in to the structures that we find from outside, more superficial, inside, more deep, inside the heart. Now the first part is kind of it's a multi-layered part. So I'm going to actually highlight both the blue and the kind of, I think this is brown structure. This is called the pericardium. Pericardium refers to around the heart. So peri, like perimeter, around the heart. Now the brown part is going to be specifically the fibrous pericardium, and I like to think of this as basically the strong, stretchy structure of the heart that basically prevents the heart from overextending. So think about this. If the heart is contracting in your chest, right, like this, the heart isn't attached to any bones. So think about when we contract our bicep, it's connected to bones, and then when we extend our bicep, the bones basically stop, and so the muscle stops stretching, right? So we have this automatic like catch where we can't hyperextend that muscle because of the bones. But think about the heart. The heart doesn't have a bone that's attached to. So when the heart contracts, the risk is when it relaxes, it would overstretch and get way too hyperextended, and that would make it really hard to contract back, right? So the fibrous pericardium prevents that from happening. So the fibrous pericardium prevents basically hyperextension of the heart, okay? Very, very important. Now that structure keeps it from hyperextending, but the serous pericardium in the blue, we've got the serous pericardium. As you can see, the serous fluid always is a fluid that prevents friction, so it promotes lubrication. So think about it. We've got two layers of the serous membrane, just like with the abdominal cavity, the pleural cavity. All these are serous fluids, serous membranes. So the one that's actually touching the heart is called the visceral pericardium. Okay, and then this part will be called the parietal. Okay, the parietal pericardium. So basically visceral, it's touching the heart muscle tissue in this case, and then we get the parietal pericardium. The main goal though is to secrete that serous fluid into this space, and it's going to promote lubrication of the heart as it is beating. Because think about it, your heart is inside your chest cavity. If it beats, it can rub up against some structures, and that could cause some pericarditis, right? Basically inflammation of the pericardium. So instead, we've got the serous fluid surrounding the heart, and it basically beats in this frictionless environment. Absolutely amazing. Now the red structure, probably the most important. This red layer of the heart is going to be the myocardium. Myocardium. And you know that that translates to muscle heart, right? So it's a heart muscle tissue. Now with the myocardium, I want you to notice a couple things. Number one, they will be striated. So they contract exactly the same, essentially, as your skeletal muscle. So when they contract, all of these lines, these striations will shorten, and all of the heart will basically come together. So all these lines come together, heart contracts like this. The other thing I want you to know about the myocardium is that it is spiraled around the heart. So what does that mean? Well, it means that this heart muscle right here would probably come around and then behind the heart and basically loop around. What does that do for the heart muscle tissue? Well, when it contracts then, instead of just contracting like this, right, it's going to contract like this, like this. So imagine you're wringing out a rag that's full of fluid, like blood, right? Well, hopefully not in that case, you'd be a serial killer. Don't do that. But if you were to twist the rag, you get more fluid out. Whereas if you just do this, you wouldn't get as much fluid out. So the heart, by twisting its muscle, will eject more blood out of the ventricles so that we can basically get as much blood as possible out of the heart per beat. Wonderful. Now, the second thing I want you to notice about the myocardium is that there are little structures connecting muscle cell to muscle cell called gap junctions. Gap junctions. This basically allows ions, the positively charged particles like sodium and calcium, to flow very, very rapidly through all of the heart muscle, basically instantaneously. Okay, it's called a syncytium, where all the heart muscle gets positive at the same time, so all the muscle contracts at the same time. And that's important because we need to coordinate this whole contraction of the ventricles, especially to get the blood out. So that is done by gap junctions. Basically, it allows the ions to flow through all of them in a moment's notice. Okay, very, very quickly. We'll get to how that actually happens here in a little bit. Then we have the inner layer that's actually touching where the blood would be flowing through. And this, as you could guess, is called the endocardium. translating to the inner lining of the heart. And it is very smooth, and it also has some projections called trabecula carne that will actually attach, if there was one like say right here, that would attach to the valve itself to basically allow the valve to open up and then close back up, open up, close back up. And that's through the heartstrings, the chordae tendineae that I am not showing here, okay? So great layers of the heart, very important. Now, I mentioned that the heart has a lot of muscle tissue. We know that therefore, if the heart muscle tissue needs blood in order to contract, we need a good blood supply to feed this. So where does the heart get its own blood supply? Well, what I didn't show you over here is that when the left ventricle contracts and goes to the aorta will have a couple branches off of it that will go into what we call the coronary circulation. So I'm going to draw just some blood vessels coming down through all of this. Okay, so here are the coronary arteries coming down like this. I'll label them here, coronary. Now you may be wondering, what does coronary mean? Well, corona refers to crown. So these vessels are basically crowning the heart, and that's what corona means, that they look like a crown, and they're actually feeding the heart tissue itself. So when the heart contracts, right, pushes blood through the aorta, as soon as it relaxes, the blood will actually seep out through the coronary arteries, feed the heart tissue, drain back through the coronary sinus back into the right atrium. So essentially, the coronary vessels are a part of this systemic circulation to your body tissues, but the body tissue itself is your heart in this case, right? So moving on from that, if you have a blockage of the coronary vessels, so if we have a blockage, okay, so say we have like a big old plaque that forms right there, if there is a full blockage there, this is what's called a heart attack. Fancy word for that is myocardial infarction. Myocardial referring to the muscle heart. Okay, so the heart muscle has an infarction, basically a blockage of blood flow. Now, this is sadly a relatively common occurrence where people will have this blockage due to atherosclerosis. Maybe they have some smoking, drinking that they've done, and it plugs up some of those vessels due to all of those highly inflammatory chemicals they're putting in their body, and it plugs those up. Now you have no blood to the heart muscle tissue itself. The heart muscle tissue begins to die, and therefore you have a heart attack. If the heart muscle dies, obviously we can't circulate blood to the body, then you die relatively quickly. So I've got a variety of videos about treatment for this you can watch in another section, but we're gonna just stick on that. So any of the blockages of the coronary vessels is called basically coronary artery disease. It can also be called ischemic heart disease. Ischemic basically means blockage, but that could be in other places too, like the carotid vessels, all sorts of things. So ischemia is basically just blockage of blood flow. Okay, now we got the coronary vessel settled. Let's talk a little bit more about how the heart contracts on its own, right? There's basically its own conduction system where the heart will beat atrioventricle, atrioventricle, atrioventricle constantly without you even having to think about it. How does that happen? Well, we know that in the heart there are two bundles of what we call pacemaker cells. I'm going to draw one about right here and then about right here, okay? Again, these are called pacemaker cells, this one being the SA node, and this one being the AV node. Now with these, it stands for sinoatrial, sinus rhythm, common sinus rhythm for your heart, and then atria, it's in the atrium. And then the AV node, atrioventricular node, it's found between the atria and the ventricle. Now what these guys will do is spontaneously, specifically the SA node, spontaneously get positive, okay? So I don't know if you know this, but you definitely should if you're taking an A&P class. When cells are just kind of doing their normal thing, they are typically negatively charged. Okay, there's a lot of proteins that are negatively charged in these cells. So the heart muscle at rest is going to be negatively charged, as well as the SA node. Now, at a certain point, SA node will actually gain some sodium ions through things called funny channels, and it will spontaneously get positively charged. Well, when things get positive, things happen, okay? So as an example here, the SA node will share its positivity through the atria in a very quick way. So all of these negatives will turn to positives. We'll draw them kind of a plus orange, kind of a black and orange because, you know, it's Halloween season right here. When the atria get positive, I want you to know that they will contract, which is awesome because we're going to push blood from atria down to the ventricles. But immediately after, we need the ventricles to contract. So those positives will be shared down to the AV node. And one quick important note, the AV node pauses for about 0.1 seconds. That is vitally important because think about it. If the AV node fired immediately, basically the atria and the ventricle would contract at the same time. But instead, what did we want? We want atria ventricle, atria ventricle. That time in between atria contracting and ventricle contracting is 0.1 seconds because the AV node pauses ever so slightly. And once the AV node sends its signal, it's going to go through a few different structures you need to know. So the first one is going to be called the bundle of His. It's very strange. Even if you are a female, you still have a bundle of His, okay? So that is that little branch coming off of the AV node. I'll write that down, bundle of His. But then it's going to branch off and go to the two different parts of the heart. So then we call these the bundle branches. I'm going to draw those here. And we've got the bundle branches next. All right, and then after the bundle branches, we're going to innervate basically all of the ventricular muscle through here. And we call the last bit the Purkinje fibers. Purkinje fibers. Okay, so that is basically all the wiring, essentially, of the conduction system. So AV node fires, goes through bundle of His, bundle branches, and then Purkinje fibers, making all the ventricular muscle very, very positively charged. Therefore, the ventricular muscles contract, okay? So again, all in all, SA node fires, all the atria contract, AV node pauses for 0.1 seconds, sends it down here, ventricle muscles contract, so we get that conduction system of da-da-da-da. Very cool. Now, if you want to look at how this actually occurs, I recommend watching the video about cardiac action potential, seeing how these cells actually get positively charged. Or if you want to know how the ECGs actually demonstrate these electrical impulses, you can also watch that video here or in the description below. But what we're going to do now is, I know you are going to be preparing for tests, right? So I want to give you a few questions that an instructor could ask you to really see if you understand these topics. All right, so here are 6 different questions that an instructor could ask you about the heart, what we've gone over so far. So let's start first with, in right-sided heart failure, what occurs? In right-sided heart failure, what occurs? Well, let's say the right side of the heart is failing. So whenever we're talking about heart failure, we're talking about the ventricles in particular not being able to get blood out of the heart sufficiently. So if the right side of the heart is failing, we will basically have a backing up of blood on the right side of the heart. What does that mean? Well, if the blood is pooling in that right ventricle, right, and we can't get it out through the pulmonary artery, what will happen is it will begin to backflow into the right atrium. When that occurs, one thing is we can actually release ANH, atrial natriuretic hormone. We won't get into that. But if we follow the path, right, if we back up into the right atrium, where does that back up into next? Well, it backs up into the vena cava, right? Especially this inferior vena cava. And you can have something called systemic edema. Why is that? What is edema? Fluid filling a space. Where? Throughout the systemic part of your body. So what's happening is the blood is backing up all the way to your body tissues, and it will begin to fill the interstitial space between the cells. That is called edema. So a lot of the times you will have swelling in the legs and the ankles because all that fluid's backing up on the right side because of gravity, into the lower extremities. Really interesting. And the same thing could occur on the left side of the heart, but in that case, we're going to back up into the lungs. It's called pulmonary edema. You can have some shortness of breath and some crackling when you're breathing. Now the next one, mitral stenosis. What's occurring? These are new terms. Mitral, meaning the mitral valve. Remember, mitral has that L, it's on the left side of the heart. And in stenosis, what's happening? We have a thickening of the valve itself. Okay, so then that makes it really hard to basically open and close properly. So once again, if we've got the stiff, thick valve, blood, when it gets down to the left ventricle, obviously we're pumping blood out, but we're having problems getting blood from the left atrium into that left ventricle. So it's getting blocked. What will occur? Once again, backflow. So we could have the same exact thing. Blood could back into the lungs with mitral stenosis, fill up the lungs with fluid, and that would cause, again, pulmonary edema. Okay, so interesting, any issue with valves as well as heart failure could personify as the same issue. Does that make sense? Okay, so we could have left-sided heart failure, we can't get the blood out, so it backs up. We could also have mitral stenosis, it also backs up. On the right-sided heart failure, or we could have tricuspid stenosis, either way backing up and having that systemic edema. So that's a really key point if you're ever asking questions or getting asked that question in an anatomy class. Okay, gap junctions function, what's that one? Well, we know gap junctions are found in between heart muscle tissue, allows ions to pass through them very quickly to create that syncytium, where we're contracting everything at the same time. So that's the function of the gap junctions. Are all arteries oxygenated? This is a really good trick question a lot of instructors give. Well, we have the pulmonary artery here, and we've got the aorta here, right? Look at the color difference. Pulmonary arteries are actually deoxygenated, whereas the aorta and the systemic arteries are all oxygenated. So no, not all arteries are oxygenated, nor are all veins deoxygenated, right? Because pulmonary vein, right, is oxygenated, and the vena cava are deoxygenated. So it just depends on which one you're talking about, so be careful with that. What's the conduction system order? We went through that already. SA node to AV node, bundle of His, bundle branches, Purkinje fibers. And the order goes, atria will contract first, ventricle will contract second. Last one, which valve will blood pass through next if we're in the right ventricle? Well, if we're in the right ventricle, we're going to go up through the pulmonary artery, we're shooting up to the moon, so it'd be the pulmonary semilunar valve. Very good. So those are some questions you could get asked. So next we're going to go into a little bit of cardiac output, as well as the cardiac cycle and circulation. Okay, so when we're talking about the cardiac cycle, we're basically saying, hey, what is happening as the atria and ventricles are contracting and relaxing throughout the heart's beat? So I want you to know two main terms. The first one is systole or systole, and that is just going to be referring to active contraction of a structure. So if I say atrial systole, it means that the atria are contracting at that point. Whereas diastole or diastole basically means relaxing at that point, okay? So if I say it's an atrial diastole, the atria are relaxing. So let's first look at this little diagram. What I've got here, it's a very simplified diagram. You can draw it yourself. We've got the right atrium up here, okay? And the left atrium up here. And then obviously we've got some valves there. So we've got the tricuspid valve on the right side. We've got the bicuspid or mitral valve on the left side. And then finally, this is just going to represent both semilunar valves. So semilunar valves of both the pulmonary and the aorta, okay? Now, that being said, when we're looking at them, we're going to be looking at when the valves open and close, as well as where the blood is going. So let's actually draw this out together if you would like. The first part, we've got both atria and ventricles here. I forgot to label these guys. So this would be the right ventricle right here. We've got the left ventricle right here. And both of them are relaxing. So this is when the heart is in total diastole. It is resting between beats. At this point, though, there will be some veins bringing blood back into the right atrium. I can kind of keep it in blue to keep it separate. And then the left atrium as well. So we're going to be filling these guys up. But as you notice, the valves, the AV valves, are open. So some of the blood is actually going to be leaking into the ventricles. This is called passive filling of the ventricles. Now, the first thing that's going to happen is that the atria are going to do what? Atria are going to contract. So we're going to call this atrial systole. But at the same time, what are the ventricles doing? Well, they're still relaxing. So we are still in ventricular diastole, okay? So we haven't injected any blood from the heart, but the atria are pushing blood down into the ventricles. So all the blood here is going to fill the ventricles, okay? So we've got really good filling of the ventricles at this point, but we want to get all that blood into the, again, either the aorta or the pulmonary artery, depending on which one we're talking about. Okay, now the moment after that, okay, this is, I believe it's called isovolumetric contraction. There is a point at which the ventricles are filled with blood, okay, but they haven't contracted yet, and the valves, the AV valves, have shut. So I want you to note that here, the AV valves, atrioventricular valves, shut. Fun fact, this is the first heart sound when these valves shut. That's the lub that we usually call the first heart sound. So really, the sounds of the heart, when you're listening to the heart with a stethoscope, is listening to the valves close. One interesting connection here is if you hear the valves close and you hear a whooshing sound, that actually means the valves didn't shut properly, and there's blood basically going backwards and forwards through the valves even though it should have shut. That's called a heart murmur. You may have heard of that before. So that's the first heart sound that you hear. It's the AV valves shutting up. Now the next part, obviously, after we shut those valves, is we have, as you notice, the atria are in what now? We have atrial diastole at both of these points. So I'm going to write atrial diastole for both of these guys because they're relaxed. But then immediately after these guys shut, what happens? We have ventricular systole. So the ventricles are going to contract, and we call this total heart systole. So if a doctor just says, hey, the heart's in systole, it's referring to the ventricles contracting. So the ventricles contract. As you notice, we push the blood up and out of the semilunar valve. So I'm going to draw both blood flow, leaving the heart, boom. Awesome. And we will have a little bit of blood kind of left over here. Not too much, but a little bit. Okay. And the atria are also going to start passively filling again, getting ready for that next step once again. Okay, so at this point, what happens? Well, we have the semilunar valves open up. Okay, atria are starting to passively fill. We've got a little blood left over in the ventricles. What's immediately going to happen next? Well, what valves shut? What valves shut after ventricular systole? We see that the semilunar valves shut, right? So I'm going to write that here. After this whole cycle, semilunar valves shut. What do you think that is? That is the second heart sound, and that is going to be the dub. Lub dup, or sometimes people say dup. Lub dup, lub dup. So the second heart sound is actually the semilunar valves shutting up after ventricular systole, okay? Now that being said, we can extend this idea. Look at the blood that left the heart. This amount of blood that left the heart is something called stroke volume, which makes sense. The heart made a stroke, a contraction. The volume is how much blood left the heart in that contraction. So interestingly, if we take that stroke volume, okay, I'm going to say stroke volume, and we multiply it by the amount of beats your heart makes a minute, which is called your heart rate, okay, and again, that's in beats per minute, we get something called cardiac output, which is the total amount of blood leaving the heart every minute, okay? A normal cardiac output is going to be somewhere around 5,000 milliliters every minute. And that equates to give or take 70 milliliters of stroke volume times about 70 beats per minute. Now, what's interesting about cardiac output, that is about a 1.25 gallons, 1.25 gallons. That is actually the amount of blood you actually have in your body on average. So every minute, your blood is circulating completely throughout your body. That is insane. Your heart is working so hard every single minute. So what I want you to notice here is something interesting. The numbers on the right obviously don't equal that. I suppose it would be 4,900 if you want to be exact. But let's say as an example, what if my heart rate is resting at like 50 beats per minute? Anybody have like a resting heart rate of 50 beats per minute? Well, think about cardiac output as a homeostatic variable. Okay, so I'm going to say homeostasis. We need to keep this constant to keep blood circulating throughout the body to keep you alive. So what's interesting is if I have a resting heartbeat of 50, my cardiac output still has to stay, we'll say 5,000 milliliters a minute. What does my stroke volume be? What does my stroke volume be? That was a terrible sentence. Well, if you can do simple math, my stroke volume could be 100 milliliters. So we say that this heart, okay, if you have a really high stroke volume, low resting heart rate, this is a very strong and efficient heart. One reason for that is if you train your heart, if you do a lot of cardiovascular exercise, your heart will get very strong, that myocardium layer. So therefore, every time it beats, it'll push out more blood. It'll push out more stroke volume per beat. Therefore, your beats per minute, your heart rate doesn't really have to be that high. It can be lower. But that being said, cardiac output is also a movable target. Let's say you start exercising and your muscles need more glucose and oxygen. The demand of cardiac output could go up to, say, 15,000 milliliters a minute during exercise. Okay, so it's not a stable point. It kind of changes as necessary, similar with blood pressure. Okay, we can kind of adjust based on your activity levels. Okay, now one thing with cardiac output that I want to briefly teach, and I got videos for it if you need to, are two concepts called preload and afterload. Okay, preload and afterload. Let me show you on this diagram what I mean by that. Preload is basically the filling of the heart before contraction, specifically filling of the ventricles before contraction. So think about it this way. If the ventricles fill up a lot, okay, it's kind of like a bow and arrow, where if you pull the bow, you pull the ventricles out, the arrow will go further. Same thing here. The more we fill the ventricles, the more blood will shoot out because the heart contracts back stronger. So I want you to say the more ventricular filling, the higher the strength of contraction of the heart. Therefore, we will have more stroke volume. Okay, so think about it. As blood comes back through the veins, fills, atria contract, push the ventricles even further stretched, the more will leave. So more preload, more stroke volume. But afterload is an interesting one. Afterload is basically saying, hey, once the blood has left the heart, there will still be blood kind of sitting upon your semilunar valves and throughout circulation. So there is a force that is actually pushing back on your semilunar valves, and we call that force afterload. We can also call that systemic vascular resistance. There's a lot of different names for that. But if your afterload is really high, say you're a very big person, you've got a lot of blood, there's a lot of force pushing back on your heart. Okay, so I'm going to call that afterload is the force basically pushing back on your heart. Now think about this. If afterload is really, really high, what do you think it'll do to stroke volume? Well, it'll probably decrease it. So more afterload usually leads to less stroke volume. And what can contribute to high afterload is a high body mass, okay, thick blood, as well as narrow blood vessels. Okay, say you have like atherosclerosis, or you've got some plaques forming on your arteries. Those narrow. Whenever you narrow a space, especially in the cardiovascular system, you increase the pressure, so you'll increase that force. So those are some things that could increase afterload, therefore decreasing stroke volume. So sometimes what's interesting, if you have of high body mass, maybe you're obese, maybe you've got some narrow blood vessels, you've got a high blood pressure, what that does is decreases your stroke volume. So what will happen to your heart rate? Well, let's say if your stroke volume, right, decreased to say 50, well, your heart rate has to compensate and get to 100 beats per minute just to meet the demand of cardiac output. So this is how, obviously, certain diseases like hypertension and like heart failure even could cause your heart rate to change to try to keep that cardiac output up. All right. Now, I've mentioned several things with blood vessels and circulation, so let's talk a little bit more about circulation throughout the entire body as well as blood pressure. All right, so in this section, we're going to go through circulation as well as blood pressure. So we're going to start with circulation, and this is physiologically accurate, but anatomically not at all. It doesn't look very nice, but I like simplistic drawings. So this is representing, obviously, the heart. The heart's going to contract, and it's going to push blood through a variety of vessels, getting to the cells or the lungs, depending on which system, and then we're going to come back through these vessels. So we're kind of going in this counterclockwise manner. Clearly, I've never read a clock before. Okay, so let's talk about some of the vessels that we're going to pass through again, regardless of whether we're on the systemic side, like the left side of the heart pumping to the body, or if we're actually pumping to the lungs or the pulmonary side. Both of these will hold true. regardless. So the first blood vessel we're going to pass through right here is going to be our elastic arteries. Now you can guess the name elastic because they are going to be stretchy, right? The heart's going to contract and the blood's going to pool into a very small blood vessel, but it's going to open up, be able to expand, right, and contract back. So we're going to go through elastic arteries, okay? Like the aorta, the brachiocephalic, these really stretchy arteries. But then we're going to go through these muscular arteries, and that's going to be a lot of them. We have a lot of muscular arteries throughout them, and they are quite jacked. Why are they jacked? Because they have smooth muscle wrapped around the outside. So if I were to do a little cross section, look on the inside, we would see a lot of smooth muscle wrapped around the outside of these arteries. Now, what that allows these arteries to do is obviously adjust their diameter. They could potentially narrow. That's called vasoconstriction. So I'll write that vasoconstriction. Or we can go the other direction. That's going to be vasodilation. So I'll say vasodilation. Now, when will they do that? We'll learn later on. Okay, so we can adjust the diameters. Next, we're going to get to third, the arterioles, which literally translates to tiny artery. Now, what's interesting about the arterioles, these small guys right here, is they also have smooth muscle wrapped around the outside. So they have the capacity to vasodilate and vasoconstrict as well. So I'll draw that. Okay, so two different blood vessels at the artery system, and they can both kind of adjust their muscle. We'll talk about why and when later. But then I want you to star the heck out of this location, the capillaries. So here we are finally at the capillaries. And the whole goal of the arterial system is to get blood from the heart to the capillaries successfully. Because this is where, obviously, if we're in the systemic circuit, we're going to take blood or oxygen from the bloodstream, give it to the cells to make ATP, and then the cells are obviously going to exchange their gas, CO2, back into circulation as well. But if we're at the lungs, the pulmonary circuit, that CO2 is actually going to leave, go into the lungs and out of the body, or we have the oxygen gas that is going to be coming into the blood. So depending on the location you are at, we're exchanging the different gas, okay? And we're also going to have waste and nutrient to exchange at this point as well. Now, once we feed the cells or bring in oxygen from the air outside of us, we're going to travel back through the venous system. The first ones will be small veins, literally translated to venules. Okay, means tiny vein. Then we're going to come through the major veins, number 5. Okay, so these will be, if I can write a 5 correctly, major veins. Now again, with the major veins, we will also have smooth muscle wrapped around the outside that will be able to vasoconstrict under certain circumstances, okay? Now I've mentioned that over and over again. These muscles can contract and expand under certain circumstances. Well, they will only do it if they receive a signal, which in other words means they have receptors. So I'm going to draw receptors on these. The first one, muscular arteries, as well as the major veins, will have these receptors called alpha-1 receptors, okay? And on these alpha-1 receptors, we can bind a hormone called epinephrine, okay? We're gonna bind epinephrine here in a second. But also at the arterioles, these arterioles also have a receptor, and this is going to be called a beta-2 receptor. Okay, so we have alpha-1 receptors, beta-2 receptors, both will bind epinephrine, but even so, not even in the blood vessels, in the heart itself. Do you guys remember that we have our SA node up here? And the SA node contains beta-1 receptors. I'm going to draw a new receptor. This will be a beta-1 receptor. And then finally, on the heart muscle tissue itself, so think about the myocardium, the muscle tissue of the, specifically the left and right ventricles, but also the atria, we will also have beta-1 receptors. Now notice the shape of all of these. They all are squares, so they're all going to be nerds, right? If you're a square, you're a nerd. That was a bad joke. They will all bind the same hormone, epinephrine. So epinephrine, if you don't know already, is going to be released in fight or flight circumstances. So you're exercising, you're working out, you're running away from something, you're stressed out, you're releasing epinephrine. When that occurs, what's actually happening is we are trying to feed the capillaries faster. I'm going to say feed capillaries and cells, obviously, the goal is to feed the cells through the capillaries faster. Well, how can we do that? How can we feed the capillaries and cells faster? Well, we could increase our blood pressure or our mean arterial pressure, right? So how do we do that? How do we raise pressure which increases the speed? Well, epinephrine. So check this out. When epinephrine binds to beta 1 receptors in the SA node, what will happen is it will increase the heart rate. It'll tell the SA node, hey, beat faster, beat faster, beat faster. So that'll increase cardiac output. If we increase cardiac output, check this out. I'm going to erase this real quick. Cardiac output is stroke volume times heart rate. So if we increase heart rate, we're going to raise our blood pressure. We'll talk a little more about that in a second. Secondly, if we bind beta 1 receptors on the heart muscle tissue itself, it'll increase the calcium amount in the heart, therefore increasing the strength of the contraction. If we increase the strength of contraction, guess what? We increase our stroke volume, so that will also increase our blood pressure. So already, the heart itself basically responds to epinephrine by contracting harder and faster, getting the blood to the capillaries as fast as possible. Again, remember, we're exercising or we're stressed. But secondly, in the muscular arteries, epinephrine will bind, and check it out. When it is bound to the receptors, what did it do? It vasoconstricted. Anytime we vasoconstrict blood vessels, we're actually going to increase the blood pressure or the mean arterial pressure we'll talk about in a second. Why is that? Well, the second variable that can affect mean arterial pressure is something called systemic vascular resistance. I'll write that out real quick. Systemic vascular blood vessel resistance. It's the resistance of flow. Now, this sounds very similar to something called afterload. Remember afterload, where is the pressure pushing back on the heart, resisting its flow? It's essentially the same thing. And remember, afterload, what could affect afterload? What can increase it? A decreased diameter of the blood vessels. So by doing that, by vasoconstricting these arteries here, what are we doing? We're increasing our mean arterial pressure. Pretty cool. And that's going to speed up the blood flow to the capillaries. That's the whole goal. Now, that being said, once we get to the capillaries, we want the blood to flood those areas. So flood the cells with nutrients and all that stuff. So what do we actually want to do to the arterioles? They're the ones feeding the capillaries. So we're actually going to bind epinephrine, and it's going to vasodilate. So it vasodilates the arterioles. That way, we're opening the floodgates to feed the capillaries dramatically more than if it was vasoconstricted. So interestingly, there's an opposite effect. Anytime epinephrine binds a 1, beta 1, alpha 1, we're always going to trigger some stimulation. But whenever it binds to something with a 2, like beta 2 receptors, we're actually going to relax it, inhibit it. Okay, that's a good remembrance. So 1 equals stimulate, and then 2 equals inhibit or relax. Okay? All right. So that being said, once we feed the capillaries, we need to get the blood back to the heart and back to the capillaries as fast as possible. How do we do that? Once again, what is on the veins? We've got alpha 1 receptors. So whenever epinephrine is bound, we are going to have them vasoconstricted. Okay? So that being said, all that epinephrine does increases cardiac output of the heart, decreases diameter of these major blood vessels, speeding up the process of getting blood to the capillaries and back, okay? So I've mentioned blood pressure a lot, and I talked about mean arterial pressure. These have these two main variables, cardiac output, systemic vascular resistance, but you may have heard of blood pressure as, say, 120/80 millimeters of mercury. And that is true, right? You can measure that because the top number is measuring the systolic value of the pressure on your arteries when the heart's contracting. So as an example, if you put your finger to your carotid pulse, you feel your pulse. When you feel that artery push against your finger, that's when the heart's actively contracting. So the heart contracts, pushes out the blood vessel, and that's your systolic pressure. Remember systole? Contraction. But then when it relaxes back into your neck, there's still a pressure on their artery. There's still blood there. That's going to be called our diastolic value. So diastolic, at rest, that is that number. But that is measured in a moment, okay? Like you go to the doctor, they measure it, they give you those two numbers. That's not an accurate representation of your average mean blood pressure throughout time. So the way we like to calculate it in A&P is through mean arterial pressure with these three variables. So let me give you an example. We have a process in your body that raises your blood pressure. By the way, let me give you a very fun fact. Fun fact. Your body is so terrified of low blood pressure that it is really good at raising it. I want you to say, body is very good at raising blood pressure. It's not very good at decreasing it. Okay, why would that be? Why would that be? Because blood pressure, again, is a measure of how well we're feeding these cells. So if blood pressure drops really rapidly, basically means we're not feeding our cells very successful and we could die very fast. So our body compensates and says, hey, no matter what, I'm going to be able to raise my blood pressure. One example, epinephrine, right? We release epinephrine to raise our blood pressure. But the main way we actually raise our blood pressure in our bodies is through something called the RAS system. I'm not going to go through the details of it, but let me give you an overview. This is called the renin-angiotensin aldosterone system. Very classy name. It makes you sound smart if you tell your parents about it. So go home and say, I learned about the RAS, renin-angiotensin aldosterone system. You'll sound smart. You're welcome. Okay. Now, what is all of this? Well, this is put into motion by your kidneys. So here's your kidney. Kidney receives a really good amount of blood supply, about 20% of your cardiac output, which is pretty crazy. But when the kidneys detect something called low filtration pressure, that's basically just a fancy way of saying just your blood pressure in general. So your blood pressure drops, filtration pressure drops. Well, the kidneys don't like that because the kidneys filter your blood. And if you've never used a filter before, you need to have a certain amount of pressure on the filter to basically get the stuff out, right? To pee it out, or to basically reabsorb back into the bloodstream. So I'm going to go through the kidneys in a different video, but the kidneys don't like low filtration because it means that we're not getting the bad stuff out of our blood. So the kidneys respond by releasing a hormone into the bloodstream called renin, literally translating to the kidney protein, the kidney hormone protein. Now, renin will go through a variety of different steps, one of which is producing angiotensin 2. Now, what angiotensin 2 does is a variety of things. It will increase ADH release at the posterior pituitary, it'll increase aldosterone release at the adrenal cortex, and it'll also vasoconstrict a bunch of blood vessels. So let's think about this. What will all of these do to MAP? Well, let's think about it. Let's start with vasoconstriction. We said that if we vasoconstrict, we narrow the diameter of the blood. What happens to afterload? Well, afterload will actually increase. Yeah? So if we increase afterload, we will increase blood pressure. You see how that works? Secondly, with aldosterone, what that does is it reabsorbs more salt and water into the bloodstream. If we have more salt and water in the bloodstream, well, guess what? We have more blood volume. We have more systemic vascular resistance, as we talked about earlier. We'll also likely have more stroke volume, more blood leaving the heart, because we just have more fluid in general in the blood. So that'll also increase our MAP. And then lastly, ADH increases water reabsorption at the kidneys. Once again, same process, raising systemic vascular resistance, raising stroke volume. So that is the mechanism by which we raise MAP with the renin-angiotensin aldosterone system. So if you think about it, all of those things, raise MAP, will help basically re-regulate, I suppose, our filtration pressure, bumps it back up so the kidneys are happy, okay? So let's finish out this video talking about some specific questions your professors could give you about these topics so that you can be successful on your next exam. All right, so to finish up today, I'm gonna give you several different questions you can actually screenshot real quick, and you can actually try to answer them yourself, but I'm obviously going to answer them for you too, so you get the right answer on your tests. And if you'd like to, you can also check out the Calendly invite for tutoring with me. It's in the description as well as in the comments. So let's get started. Again, pause the video if you want to answer them yourself first. What would increased venous return do to cardiac output? Let me give you the answer first. and it would increase cardiac output. Now, the reason for that is increased venous return. We're filling the heart more if we have more venous return, bringing blood back to the heart. And if we do that, we fill the heart, and what does that do? Well, we know that it increases preload. An increased preload leads to higher stroke volume, therefore higher cardiac output. Now, this is actually the major mechanism when you're exercising. You're contracting your muscles, compressing the veins, forcing blood back through those valves, one-way direction, back to the heart. So that way, obviously, we increase venous return, increase preload, increase cardiac output. The second thing, what can epinephrine affect to raise MAP? Well, we know it can act on the beta 1 receptors, that is in the heart muscle tissue, as well as in the SA node to increase heart rate, right? We can also act on the alpha 1 receptors, that is on the arteries, the muscular arteries and veins, and we are going to vasoconstrict them and therefore increase blood pressure. So both of these, right, will increase blood pressure by acting on those receptor types. The third one, which is worse in the extreme, high or low blood pressure? Think carefully about this one. A lot of people get this wrong. Low blood pressure will always be worse in the extreme. Hypotension, acute hypotension, that is always going to kill you because, again, blood pressure is a measure of how well blood is circulating to the capillaries. So if we have literally like no circulation to the capillaries, you are going to die very rapidly. Now, that being said, high blood pressure is also very bad, hypertension, and that's why we treat it with blood pressure medications because it can long-term cause strokes and aneurysms and all sorts of bad things like atherosclerosis, coronary artery disease. So we want to get that managed, but in the extreme, in the moment, low blood pressure is always worse, which is why your body's so good at raising it. Now, what's the thickest layer of the heart? This should be a relatively easy one. This is the myocardium. We drew that earlier, and that's the muscle layer. It's very thick and strong. And which side is thicker, the left or the right side? Well, you can probably guess the left side of the heart is much thicker because it has to pump to the whole body, whereas the right side just has to pump to the lungs. So if you actually do a dissection of a heart, you can feel the left side of the heart is really tough and strong because it's really thick, and then the right side's a little more flabby, just pumping to the lungs. Now, where is blood traveling slowest in circulation? We talked about that earlier. That is the capillaries. And again, with the capillaries, we want to slow the blood down so we can slowly exchange, like the toll booth, exchange the gases and nutrients. If we went too fast, that wouldn't work very well. What would happen to MAP if we had 3 different scenarios? So we're going to talk about sympathetic, parasympathetic first. The parasympathetic is going to be designated as this orange, and it originates in this case in cranial nerve 10, the vagus nerve. And it will actually go in and talk to the SA node. Okay, SA node controls heart rate. Okay, so if we're talking about MAP and heart rate increases MAP because heart rate's in cardiac output. So you got to go back to that calculation. Now, the parasympathetic neuron, the vagus nerve, is going to send inhibitory neurotransmitters. In this case, this is acetylcholine. It will actually decrease the heart rate by basically inhibiting the SA node, therefore basically inhibiting MAP, decreasing mean arterial pressure. Whereas on the flip side, sympathetic stimulation, the red originating in the thoracolumbar region of the spinal cord, we're going to send a stimulatory neurotransmitter. In this case, it is norepinephrine, okay? And that will actually increase your heart rate, therefore increasing mean arterial pressure. Okay, so they're kind of acting on the same structure, but doing 2 opposite effects. And then the last one, ADH antagonist. Anytime you see antagonist, we are opposing whatever we're talking about. So remember, ADH increased water back into the blood, which therefore increased our stroke volume and therefore increased our mean arterial pressure. It also increased systemic vascular resistance, if you remember that as well. Okay, so obviously ADH does that, but we are antagonizing it. Therefore, the reverse will be true. There will be less water reabsorbed. You'll actually pee out more, interestingly enough. We'll have decreased stroke volume, decreased SVR, and we'll have therefore decreased mean arterial pressure. So a lot of instructors that I know give these sort of, hey, if this, then what questions. So what if you had like an aldosterone agonist? Well, that means we are amplifying the effect of aldosterone. We would actually increase salt and water reabsorption, increase blood pressure. So be careful about antagonists and agonists. Hey, that's all for today. Real quick, if you want to check out more about the cardiovascular system, check out this playlist. Again, check out that link, and thank you for watching. I hope this was helpful.